Hello, my name is Woojin Jeong, and today I'm sharing the findings from studying how adolescent THC exposure causes dimorphic alterations in the response to viral infection in mice subjects based on sex. From previous research, it was found that the microglia plays a huge role in the long-lasting effects of abused drug usage. Adolescent THC exposure in models of mice caused pervasive and persistent behavioral alterations due to the dysfunctional microglia. Furthermore, adolescent THC exposure alters the transcription of genes in adult microglia that are involved in the homeostasis and innate immunity of the microglia. Summarizing the preliminary findings, the new question we had was, how does this THC exposure in the adolescent brain of mice subjects with viral infections alter or affect the immune response and behavioral properties? In an attempt to answer this question, we set up a hypothesis stating that prolonged exposure to THC used during adolescence might cause changes in the molecular function of microglia and the alteration of gene transcription, which persistently alters sickness behavior and brain-wide gene expression changes associated with responses to poly-IC. For the research method, we used both male and female mice subjects and separated them in ventilated cages based on sex with free access to food and water. They were also divided by control and treatment cohorts to compare the differences. THC, poly-IC, and other required chemicals and solvents were purchased from appropriate domains. THC and poly-IC were given to the mice by intraperitoneal injection for a span of a few weeks with their appropriately measured concentrations. For data, the baseline temperature and post-injection temperature were measured through implanting thermosensitive microchips. Also, the open field test was performed post-injection for each mice to analyze their cognitive and behavioral responses. Afterwards, whole brain tissue of the mice were collected and the isolated RNA samples were analyzed. Moving on to the results. Adolescent THC significantly blunted the poly-IC-induced increase in body temperature change in female mice, while exerting weaker effects in males. From analyzing the poly-IC-induced sickness behavior, it is shown that the effects of poly-IC and adolescent THC are significantly related to sex. From analyzing the transcription of immune-related genes, poly-IC significantly enhanced the transcription of 527 shared genes for the control group, where a less significant effect was observed with the THC-treated mice with 323 shared genes enhanced. Also, in male adolescent THC mice, poly-IC enhanced expression of genes enriched in geocategories such as cellular response to cytokine stimulus, while no such enhancement was observed in the female cohort. Lastly, the poly-IC regulated transcription of neuron-related genes were analyzed, showing significant suppression of the transcription genes implicated in neurodevelopment and synaptic transmission in the brain of male control mice, while not female. It is analyzed that early lab exposure to THC prevented this effect, as shown by the lack of response to poly-IC in adolescent THC cohorts. In conclusion, the results in an analysis of the behavioral study and poly-IC regulated transcription of genes shows sexually dimorphic alterations of the responses. Adolescent THC exposure has reduced body temperature and caused weaker antiviral gene response for female mice, while it caused weaker neural gene response for male mice. Both sex showed no motor improvement from the THC treatment. Based on these results, future studies will be focused on exploring the specific mechanisms of the interaction between adolescent THC exposure and poly-IC, as well as examining if pubertal factors play a role in the impact of THC administration on mice. Thank you very much.